Hi, my name is Effie Nikki, and today we're going to talk about hemostasis. Hopefully by now you've been doing a little word association and you figured out hemo means blood. You may or may not know stasis, but it means halting. We think of it as stopping. I have highlighted here in red a basic definition. Hemostasis is a fast series of reactions for stoppage of bleeding. It's fast, it's localized, it's controlled. Every day we have tiny little tears in our capillaries, in our arteries, in our veins, and if we have to have a controlled, fast, localized process in order to be able to stop that bleeding. So right now we're undergoing hemostasis, we have clotting going on. Sometimes we think of this more just happening in the large, major trauma, but this is happening every day. Right now as we're watching this recording, you're undergoing this process. Okay, so I've outlined the three major steps here. We're going to look at number one, the vascular spasm. So these are a series of three events that are going to occur to stop bleeding. So the very first one, vascular spasm, you're going to have some sort of vasoconstriction. So vaso means vessel, constriction means tightening or making smaller. So the damaged blood vessel is going to undergo vasoconstriction. This is going to stop blood supply to that vessel temporarily so that you can do steps 2 and 3 that we'll look at in just a second. There are some chemicals that are released by damaged endothelial cells. Hopefully you know what endothelial cells are, but if not, I'll just recap real quick. Endothelial cells line the heart and all of your blood vessels, pretty much your cardiovascular system. They're just a subset of simple squamous epithelial cells. So if you don't remember your histology, just go back quickly and look at simple squamous epithelial cells. They're good for diffusion of small substances. We just say endothelial because of its location. So endothelial, it's a subset of simple squamous epithelial. That's it. Okay, back to the chemicals. The chemical that's released is called prostaglandin. So when you have a damage or a tear to a blood vessel, vasoconstriction is going to happen. We're going to limit the blood supply, and we're going to see chemicals that are going to be released by platelets and by the endothelial cell layer inside the blood vessel. And these prostaglandins are like signals. They say, hey, we have damage here. We need some help. Come to this area right now. And we'll see how this leads to number 2, which is the platelet plug formation. So this is where we also use the word aggregation. So you have platelet aggregation, sticking together of platelets. So let me back up just one second. Platelets don't normally stick to endothelial cells. Right now we have blood moving through our system. Platelets are in there. They're not sticking up against everything that they see. It's because the endothelial cells are releasing something called prostacyclin. And I know it sounds like prostaglandin, so let's not get confused. So it's a prostacyclin. It's released by the endothelial cells, and it doesn't allow platelets to stick. So when the endothelial cell is damaged, it's not making any more prostacyclin, right? And the ones that are around where that tear was start releasing prostaglandin. That prostaglandin recruits platelets to come to that area and plug up the hole. That's where we get platelet plug formation. So you can see in the third line here, the platelets are going to swell, they're going to aggregate or come together, they're going to swell, they're going to become spiked, they're going to become sticky, and they're going to release chemical messengers which cause more platelets. Again, that's the prostaglandins. And this is an example of a positive feedback mechanism. We mentioned this way back in chapter 1 of 201. So if you want to go back and read about a positive feedback and negative feedback was the opposite. Positive feedback, you're driving in one direction, so platelets come and stick to the tear. More platelets are sent to that area. So it's a positive feedback. It keeps driving in a forward direction. Till eventually the platelets plug the hole, or the platelets plug the damaged blood vessel. The third step is called coagulation. This is going to help reinforce the platelet plug. We're going to have something called fibrin, these threads that are going to come in, and their job is to pretty much turn your liquid blood into more of a gel-like substance, so all of the factors can come together that are involved with healing and actually heal the broken blood vessel. This is a multi-step process. We're going to see many clotting factors on the next slide. Don't get too freaked out. Most teachers are not going to make you know all of the clotting factors, but check with them before I say that. We'll talk about the last couple of steps in the coagulation, and those are really important. But all of these clotting factors that I'm just getting ready to show you, most of the time you just need to know generally where they come from and why they're important. Okay, so this is showing you two different pathways, which I'm going to come back and talk about. We see intrinsic pathway and extrinsic pathway at the top. For right now, just forget about it. Look at all the clotting factors. So everything you can see here with the Roman numeral is a clotting factor. Let me get this pen working. Come on. Okay, so here is clotting factor 12. It is inactive in this form, but when it gets activated, can I spell? Okay. When it becomes activated, it's now 12A. What does this activated clotting factor do? It turns clotting factor 11 into 11A. It went from inactive to active. So when a blood vessel is damaged, when the platelets stick to the underlying connective tissue, it's going to cause this factor 12 to be activated. So it's kind of a series of events. They call it a cascade. So one step leads to the next step, which leads to the next, which leads to the next, like a waterfall. So eventually we're going to get down to this bottom thing, which is called prothrombin activator. A couple things I want to point out. One, most of these factors are made in the liver. And they call these clotting factors. Sometimes they just say factors. Most of them are made in the liver. Several of them require vitamin K in order to be activated. Also, you can see here calcium. Hopefully you remember way back again in the beginning of 201, we talked about the need for controlled levels of calcium in your blood. We talked about your bones storing calcium. We talked about why calcium was important in muscle contraction. Well, this is just another example of the amount of calcium in your bloodstream is important because it's a factor in clotting. And if you don't have calcium at this step, nothing under that step, nothing below that step is going to occur. So calcium is also very important in this clotting factor. Also that these, I don't know if I mentioned this just a second ago, so I'm going to say it again, these are all inactive, floating in your bloodstream right now. So all of these clotting factors we're making, they're in our bloodstream, they're just in an inactive form. It's not until they're activated by platelet aggregation and the release of prostaglandins and other chemical messengers that we start activating these clotting factors, which will eventually result in a clot. Okay. This is phase 2 and 3. It says 3 phases of coagulation. Technically, what we just looked at would have been the first phase. We're going through making all these clotting factors activated till we got to that prothrombin activator. The second and third part is what we're going to look at right now. So the first thing that's going to happen is prothrombin activator is going to do what? Activate prothrombin. I like it when they name things to where it makes sense, right? So prothrombin is going to be converted into thrombin. Prothrombin activator is the cause of that. That's what's going to cause prothrombin to be turned into thrombin. Then thrombin is going to be cause fibrinogen to form this fibrin mesh. We're going to see a cross-linking occur. We're going to see the beginning of a clot formation. And remember, coagulation is turning liquid into a gel. So let's look at the flow chart. I know sometimes the wording gives us some issues. This is also going to strengthen that platelet plug formation. Okay, so this is a flow chart. This picture you do need to understand and know. I personally ask questions about this flow chart, so I would make sure that you understand it. Again, here, these are phases 2 and 3. So here's our prothrombin. Here's our prothrombin activator. It causes prothrombin to become, or the activated form of thrombin. Then thrombin is going to cause fibrinogen to become fibrin, and fibrin eventually to become this fibrin mesh, which is what I'm trying to show you in this picture right next to it. It's showing you red blood cells. It's showing you fibrin, which are fibrous tissues, or fibrous threads is probably a better word, and those threads are going to form a mesh. It's almost like a bed, and in that bed you're going to allow clotting factors to stick, other proteins that we'll talk about in a second, factors that are going to allow you to heal this wound. So this mesh is necessary in order for everybody to be where they need to be to heal this blood vessel that's been injured. Again, you can see calcium is important here. This is another factor, factor 13. This is going to cause fibrin to become that fibrin mesh. It actually helps cross-link the threads of fibrin. And again, fibrinogen and prothrombin are in your blood right now. They're just not activated. So these, you could say, are inactive, and then these are active forms. Okay, so this was the pathway that was directly above it. I just wanted to clarify. This was phase 1. What's the goal? The goal is to get to prothrombin activator. And then once we get to prothrombin activator, we can take prothrombin and turn it into thrombin, and then have thrombin turn the fibrinogen into the fibrin mesh, the one we just looked at. So I want to go back and talk just briefly about the top part that I said I'd get to, intrinsic pathway and extrinsic pathway. So you can see right here, extrinsic, intrinsic pathway. I'm going to show you some words on the next slide, then we're going to go back to this picture and look at it again. Okay, intrinsic pathway. What's important on all of this slide here? Intrinsic pathway, it's triggered by negatively charged surfaces. Intrinsic means it's within the blood. So within the blood is the factor that's going to activate the cascade. It's triggered by negatively charged surfaces. Activated platelets are negatively charged. The collagen that the platelets stick to are negatively charged. So that's all part of the intrinsic pathway. Also, glass will trigger the intrinsic pathway. And what's the end result of both of these pathways? Fibrin mesh, I'm making up new words, fibrin mesh, which is going to be clotting. So both of these pathways are going to result in clotting. Glass, the reason I brought that up, if you ever go to the hospital and you have blood taken and they put it in a glass tube, there has to be some sort of anticoagulant in there, something to stop the clotting process. If you just take blood and put it in a glass tube and leave it there, it's going to start clotting because the platelets are going to stick together. We're going to follow this intrinsic pathway. We're going to have all those clotting factors activated. We're going to have prothrombin activator activated, and then we're going to lead to that fibrin mesh and that clotting. All right, the second part is intrinsic pathway. This is another pathway that will end in prothrombin being activated and eventually that fibrin mesh. This one is due to a cell. If a cell is damaged, it will release something called tissue factor. The reason this is called extrinsic is it's not something that's inherent in your blood. It's something from outside of your blood. So the cell has to release this tissue factor on its cell surface, and then the cascading effect will occur, and eventually you'll get to prothrombin being activated. So back to this picture again, and I just gave you examples off to the side. So it says if a vessel ruptures, platelets cling to the collagen because it's negatively charged, right? In the underlying tissues, it'll release something called PF3, and they show you PF3 right here. Come on, do what I want you to do. There we go, PF3. PF3 is going to be released. All of these factors are going to be activated, and the end result is prothrombin activator being activated. Then we're going to go have prothrombin turned into thrombin, eventually till we get to the fibrin mesh, right? Okay, what about this one right here? When a cell is damaged, what's the cell going to do? The cell is going to release tissue factor. That tissue factor is going to cause this cascade to occur, and then eventually it's going to cause prothrombin activator to be activated. So both of these pathways lead to prothrombin activator being activated. They're just different ways for them to occur. And you can see by the number of factors here, this is a very fast pathway, and this is a very slow pathway. This pathway on the left has many, many, many steps, where the pathway on the right has fewer steps. Usually both of these are occurring. Both can occur at the same time. It's not like, oh, you have one or you have the other and you don't have both. Sometimes you're going to have damage where you're damaging the cell and it's going to expose tissue factor and it's going to follow the extrinsic, and then sometimes you're going to have damage to the blood vessel at the same time. So both of these pathways can be occurring. Just realize there's two separate pathways. They all converge on prothrombin activator. Okay, clot retraction and repair. We don't want that clot to hang out forever. Remember I said we have multiple blood vessel breakage or leakage, things that have to be repaired over the course of a day. If every clot stayed there and wasn't eventually removed, we would have a problem. We'd eventually have an occlusion. We'd have tons and tons of clots that would block up that capillary, and it'd be no good to us. So we have to be able to retract the clot, which is seal the gap, bring the two edges of the blood vessel together, and we also have to get rid of that clot eventually, the hardened blood meshwork, right? So the first thing here is going to talk about clot retraction. We have contractile proteins called actin and myosin. Hopefully you remember these from when we did muscle. They're in the platelets. So the platelets have contractile proteins. And these contractile proteins are going to be activated within the first hour. And what's going to happen is, remember those fibrin strands? The platelets are going to pull on the fibrin strands and pretty much bring the edges of the damaged blood vessel together so that they can be healed. I often think about this as like you're tying a shoe. So the shoelaces are the fibrin, and pulling on the shoelaces causes the shoe to get tighter. Same kind of concept here. You're taking the fibrin strands, you're pulling on them and bringing the edges together so that they can be healed properly. And then the last thing here is talking about the repair. We have something called platelet-derived growth factor, and then we have, at the last item here, vascular endothelial growth factor. So do we want to repair the smooth muscle? Yes, because remember, blood vessels are made up of smooth muscle, connective tissue, and then we have that inner lining of endothelial cells. So we have to be able to repair all of those. So we have two separate growth factors. The PDGF is going to help with the smooth muscle rebuilding, fibroblasts, which are going to help rebuild the blood vessel wall, and then your VEGF, or vascular endothelial growth factor, is actually going to help those simple squamous epithelial cells reline the blood vessel so you have that nice smooth surface for your blood to flow through. Okay, this is the last part. I was talking about the clot actually being removed. So we have to do fibrinolysis. We have to lysis break the fibrin that we have created. So pretty much we want to get rid of that clot so it doesn't block the blood vessel. And again, there's another little pathway here that has to be followed. Plasminogen, which is present in the clot. When the fibrin mesh trapped the debris or the proteins and the clotting factors and all the stuff that was in your blood, it got trapped in that fibrin mesh. Along with it was something called plasminogen. That plasminogen is going to be turned into plasminogen is going to be converted to plasmin by something called tissue plasminogen activator. And you can see here there's another activator with the name in it. So what does tissue plasminogen activator do? It activates plasminogen to turn into plasmin. This plasmin is a fibrin digestive enzyme. It's going to come in and kind of chew up the fibrin that was helping hold that fibrous mesh together. But this is only going to happen after the healing process has occurred, right? You'd be defeating the whole purpose if we start getting rid of the mesh before the actual healing event. So this is at the end when you have a scab, you have that clot, and it falls off. It's because the process has healed, the plasmin is going in and digesting the fibrin that's holding that clot, and then your scab falls off. I wanted to mention the tissue plasminogen activator. This is something we've actually been able to create in a lab. We have other cells that will grow human tissue plasminogen activator, and then we can actually give that to patients to help dissolve clots. So, and that's exactly what this does, right? Tissue plasminogen activator activates plasmin, which is going to digest those fibrous or fibrin can't talk right now. Digest those fibrin threads, and that's going to cause the clot to dissolve. So we actually give patients, TPA, stroke victims, anybody that has a clot, to help break up and leak. Okay, so let's talk about this last thing, limiting our factors that limit clot growth and formation. Basically, we don't want a clot to get too large or to grow out of control. So we have to have some factors where if a clot is persistent, we have a way to stop the clot from being formed. The first thing here talks about blood flow. We want our blood to be constantly flowing so that clotting factors can't get stuck in a certain area and start activating it. The second thing here is we have some enzymes, we have some proteins that are enzymes that can stop the clotting process if we need it to. So you can look at some of these here and figure out what they are. Antithrombin, it's going to stop thrombin from turning into that fibrin mesh, right? So it stops the activation of thrombin. Protein C is going to stop several different clotting factors. It binds to the clotting factors and stops the clotting factors from becoming activated, that long cascade that we looked at. Tissue factor inhibitor. Tissue factor is released in that extrinsic pathway by the cell. So if you stop tissue factor from being released, you don't have that cascade, you don't have prothrombin activated, you don't have a clot. And then this last one here, prostacyclin, if you remember at the beginning of this lecture, I said what stops the endothelial cells and the platelets from sticking together? That the endothelial cells produce something called prostacyclin, and that stops platelets normally from sticking to endothelial. If we increase the amount of prostacyclin, the platelet can't stick to the endothelial cells even if there is slight damage there. So if we want to stop the continuation of that cascade and we want to stop a clot from getting too large, there are some enzymes that we make that allow us to limit the ability of a clot to form. I think that's it. Yay! All right, well, I'm going to do the next lecture when we talk about some of the disorders in hemostasis. So I'll see you in just a minute. Bye.